Welcome to the Biomics YouTube channel. It is a good strategy to prepare with a plan for CSIR NET JRF Life Sciences exam. To help you make your strategy, this video gives you insight of important topics asked in CSIR NET JRF exam. CSIR syllabus is vast and has questions from 13 units which covers almost all domains of life sciences. All the 13 units in CSIR have equal weightage and equal number of questions are asked from all the units. As a smart strategy, it is important to primarily focus on important topics rather than studying all the topics. In Section A, maximum 15 questions can be attempted out of 20. Whereas in Section B, 15 extra questions, and in Section C, 50 extra questions are provided. Therefore, having a planned strategy helps score maximum. We are covering Section B and Section C, as Section A is basically aptitude-based. On an average, 6 questions are asked from individual units in Section C. Therefore, all units are given almost equal weightage, however with time topics of interest vary. In Section B, each unit contribute 4 questions. However, most of the questions have been asked from molecular biology and plant physiology. Let us start with unit-wise important topics which we can focus to prepare for CSIR NET JRF Life Sciences exam. First unit is biochemistry, and it forms base of all other units. Understanding of this unit helps in clearing the concepts of other units. To cover biochemistry for CSIR NET JRF, we need to cover these topics as priority. About 4 to 5 questions are asked in Section B and Section C from biochemistry. Important topics include peptide bonds, protein structure, enzyme inhibition, enzyme kinetics, enzyme regulation, protein cleavages by trypsin, pepsin, and chymotrypsin. Additional to this topics, biomolecules stabilizing interactions, covalent and non-covalent bonds, sugar puckering, DNA, Ramachandran plot, lipids, and its relation to plasma membrane, oxidative phosphorylation, and all metabolic pathways have higher frequency of appearance in exams. From exam point of view, the most important part is glucose-related metabolism and Ramachandran plot. In past years, conformation of DNA is among focus topics. Lipids are mainly information-based and have questions related to structure and stability of plasma membrane. After biochemistry, cell biology is common for core plant sciences, animal sciences, microbiology courses, as well as for the students with technical course. Unit 2 comprises cell biology. About 4 questions are asked in B part, like cytoskeleton or extracellular matrix. Almost 6 questions are asked from cell cycle, membrane potential, and cancer in the Part C. To cover this unit, multiple topics become important to cover the syllabus. The topics include cell cycle regulation, cell junction, cellular respiration, extracellular matrix, apoptotic genes in C, elegans, ion channels including ligand-gated, voltages-gated, and mechanically gated channels, membrane potential. Further, microtubules and its function, plasma membrane composition, protostome and deuterostome, role of protein 53, retinoblastoma and MDM2 or HDM in cancer, secretory pathways of protein trafficking and techniques, and active transport microtubules are also asked. Among these topics, plasma membrane composition and protein 53 including protein transport should be actively focused for a better score in the exam. Cell cycle checkpoints, cell-to-cell fusion, cell cycle stages, CDKs, cyclins, electrical potential of a cell are frequent. But more focus should be given to CDKs and cyclins from exam point of view. Unit 3 is molecular biology, which is base for genetics and techniques as it has most of the important topics. It not only helped to clear CSIR but interview as well. The topics to be considered to cover in Unit 3 are replication, DNA damage and nucleotide excision repair mechanisms, proofreading activity of DNA polymerases in eukaryotes as well as prokaryotes, transcription, translation, gene regulation, and lac operon. DNA damage and repair forms basis of most questions from Unit 3. Further, phage control, riboswitches, all types of RNA processing, small silencing RNAs like microRNA, siRNA and piRNA, aminoacylation of tRNA, tRNA identity, aminoacyl tRNA synthetase, CDR and AIM-type cells are frequent topics. However, RNA processing or post-transcriptional modifications are important. The Unit 4 mostly includes immunology part and cell signaling. 1 or 2 questions are asked in cell signaling, but most of the questions asked from this unit belong to immunology part. Cancer-related topics are also related to signaling, so questions asked contain signaling and cancer. It is advisable to stay updated with latest research rather than focusing on outdated study materials. This unit involves cell adhesion molecules, which are one of the most important topics for CSIR. The broad topics asked from this unit are hypersensitivity types, G-protein coupled receptors, immunoglobulin functions, and digestion with papain, trypsin and mercaptoethanol, pathogens, bacterial and viral diseases, plant pathogen interaction and stress responses. Here hypersensitivity, GPCRs, and stress responses in plant are must to cover for added advantage in the exam. Few more topics from microbiology like antibiotic susceptibility testing for enteric bacteria, antibiotic-resistant bacteria, and disease related to gram-positive gram-negative bacteria can be focused. Also, topics like epitopes and paratopes, antigens, pathogenic diseases should be briefly covered to attempt information-based questions. To score in CSIR well, it is really important to cover developmental biology. In developmental biology, new research is not frequent, so questions asked in CSIR are modified and limited. To score a better rank, preparing developmental biology gives extra edge. The important topics are autonomous and conditional specification, which kind of must in developmental biology, capacitation in mammals, cleavage pattern, different larval forms in animal kingdom, egg types, symmetry of egg, extra-embryonic membranes in human developments, fertilization in sea urchin, and gastrulation in xenopus, genes involved in floral organ identity, ABC model, and floral meristem identity, regeneration in development, morphogens like retinoic acid, amphibian metamorphosis, axis development in Drosophila, gastrulation, cleavage pattern are equally important. Basically covering Drosophila and C. elegans development could be worth in further research as well as for the examination. Some topics in the plant physiology unit have higher frequency from which questions are asked. In this part, new updates are limited, so it's really nice that you prepare plant physiology and development biology properly to ensure a good score. Your earlier units, biochemistry and cell signaling, will play vital role to have good concepts from this unit. This unit covers several important topics related to plant biology. Firstly, enzymes involved in the C3 and C4 cycles, as well as their regulation. Next, the location of photosynthetic pigments such as photosystem second, photosystem first, chlorophyll, and bacteria chlorophyll, as well as their distribution within plants. Moving on, nitrogen fixation, the mechanisms of phloem loading and unloading, photomorphogenesis, and the absorption of light by various photoreceptors such as phytochrome A, B, C, D, E, cryptochrome first and second, and phototropin first and second are amongst important topics. Furthermore, the photoprotective mechanisms employed by plants to shield themselves from excessive light. Another important topic is photorespiration. The concepts of pluripotent, multipotent, and totipotent cells are important to be covered. Additionally, secondary metabolites, specifically terpenes and alkaloids, signaling pathways and the functions of different phytohormones, including auxins, ethylene, and gibberellins covering various aspects of plant growth and development are important. System physiology of animal could be optional for the students who had core plant sciences course, as both Section B and Section C has sufficient optional questions. However, this unit is easy to remember the concepts once studied. So covering few topics may help attend 1 to 2 questions in Section B and few questions out of 6 in Section C confidently. Animal physiology is very vast subject and also based on previous year papers. Not much topics are important in this part. But consider covering nephrology and heart, being easy and with less confusion. To cover this unit, these topics can be focused. The unit has few key topics that are important and relatively easier to understand. Endocrinology, reproductive biology, differences between amniotes and non-amniotes in terms of their reproductive strategies and excretory system. With base from plant and developmental and molecular biology, most of the topics of inheritance biology are important. In this unit, reading and practice become important to solve questions. Topics like pedigree and mutations in this unit are special. In the unit of inheritance biology, it is crucial to have a good understanding of various topics to excel in the CSIR exam. Reading and practicing questions are particularly important for this unit. Some of the key topics to focus on include complete and partial linkage, fluctuation test and replica plating experiment, gene interaction, genetic drift, Loxp and Cre recombinase, maternal inheritance, microbial genetics, conjugation, transduction, and transformation. Further mutant in Drosophila, ordered tetrad and octad analysis in Neurospora crassa, pedigree analysis, point and frame shift mutation, polygenic inheritance, transposable elements, phage gene integration in lysogenic and lytic cycle of E. coli genome should be covered in Unit 8. Agents involved in point and frame shift mutation, allelic interactions, dosage compensation are equally important. By focusing on these topics and dedicating time to reading and practicing questions, you will develop a strong understanding of inheritance biology and be well prepared for the CSIR exam. This is again vast unit like animal physiology, but to save your time in the unit diversity of life forms. Important parts to grab from the Unit 9 are plant and animal classification hierarchy, comparison between eukaryote, archaea, and prokaryote, different larval forms from helminths to echinoderms, food materials of green, brown and red algae, food stored in green, brown and red algae. Mostly the unit is memory-based, however covering larval forms of helminths to echinoderms would be beneficial. Unit 10 is very interesting for CSIR as well as your other studies. In this part, a good imagination is helpful to solve questions. Like development and plant physiology, it is an important unit for scoring and clearing CSIR. There are several important topics that are frequently asked in exams and require a thorough understanding. However, key concepts to cover from exam point of view are biological species concepts, endangered species, endothermic and poikilothermic animals, factors blameable for species extinction, inclusive fitness, K and R strategy, kin selection, and mode of natural selection. Further, to cover the ecology unit require a solid understanding of population ecology, population regulation, top-down ecosystem control, adaptation in animals and plants, community ecology, and different types of mimicry to avoid predation. Additionally, ecosystem, reciprocal altruism and Lotka model should also be briefly covered. By thoroughly studying and comprehending these topics, you will be well prepared to answer questions and demonstrate a strong understanding of the concepts in the field of ecology. In the unit of evolution and behavior, there are several fundamental concepts that are crucial to understand for the CSIR exam. Firstly, well-discussed Miller-Urey experiment, coacervates, protocells considered to be precursors to modern cells are among the important topics. Understanding the major steps in the origin of modern cells is essential to grasp the complexity of cellular evolution, theories of Lamarckism and Darwinism, the evidence of evolution, including fossil records and comparative anatomy. The concept of natural selection, including different modes such as directional, stabilizing, and disruptive selection in populations is also important. By comprehending these important topics, we will gain a solid foundation in the unit of evolution and behavior essential for success in the CSIR exam. In Unit 11, Drift and Bottlenecks, Genetic Drift, Punctuated Equilibrium and Phyletic Gradualism are important theories in the field of paleontology and evolutionary biology, so these should be covered. The geological time scale including chronology of events such as the formation of continents, the appearance and extinction of species, and major geological processes. Phenograms and phylogenetic trees in evolutionary biology and systematics are also important. Evidence of evolution, speciation, and evolution tree are most important and are mostly asked questions from this unit. In Unit 12 and 13 most of topics are related to cell biology, biochemistry and molecular biology techniques. Being most scoring and having analytical questions require more effort than other units. Application of other units fall in these two units. Studying the topics of restriction enzymes digestion, T plasmid, and adaptive modifications in cold-warm climate animals, as well as cold, drought, and salinity stress in plants, is crucial in the field of applied biology. Understanding restriction enzymes digestion helps in manipulating DNA for various applications, such as genetic engineering and DNA sequencing. T plasmid is important as it is commonly used in the introduction of foreign DNA into plants. Additionally, comprehending the adaptive modifications in animals and plants in response to different environmental stresses is essential for developing strategies to enhance their survival and productivity in changing climates. Unit 13, Methods in Biology, is an incredibly important and scoring unit in CSIR Life Sciences exam. This unit focuses on various techniques and methods used in biological research, which are essential for conducting experiments, analyzing data, and drawing meaningful conclusions. And quite logical and simple questions are asked to assess the understanding of the scholar taking exams. Most of the questions are related to cell biology techniques such as flow cytometry, FRET, microscopy, estimation of gene expressions by different techniques. With recent researches, CRISPR-Cas9 system should also be covered as it has higher probability to be asked. Whatever we have mentioned, these are based on previous year papers and student experience. You need to score well, revise these topics properly and cover important units such as biochemistry, cell biology, molecular bio, genetics, immunology or cancer biology, plant physiology, developmental biology, and ecology. Thank you for watching. Your suggestions and queries are invited. Please feel free to connect with the Biomics team in case you need any help or guidance.